Thank you so much, Jan, for that lovely introduction, and thanks to you and Purina for inviting me to speak today. So, like Jan said, I'm not a gastroenterologist, I'm not a nutritionist, I'm a pharmacologist. So hopefully I can bring a little bit of a different perspective to this theme of gut microbial metabolism that we've kind of been developing today. Um, and I'm really grateful. Like I said, I'm not a gastroenterologist, but I'm really grateful to work with some wonderful people here at the University of Illinois, including Dr. Barco, who have really piqued my interest in this area. So as hopefully we've presented to you over the past 2 days, we know that drugs can have a major impact on the body's different microbial populations, antibiotics being the best characterized in our veterinary species, having a major impact on microbial diversity in numbers, and also on their biotransformational function. There have been other drugs, and the list is increasing every month with every publication of other drugs and whether or not they have a major impact on the gut microbiome. Proton pump inhibitors have been looked at in both dogs and cats. They have a major impact in people on microbial diversity, not quite as much of a big deal, at least based on the information we have right now in our veterinary species. Steroids we talked about earlier this morning, and then some other drugs, phenobarbital, anthelmintics, chemotherapy, and then antifungals, which is something that Dr. Barker and I are working on right now. But it's important to remember that this is a two-way street, that it's not just the drugs that can impact microbial communities, but those microbial communities can actually impact the drug pharmacokinetics and drug pharmacodynamics. And the study of this concept is called pharmacomicrobiomics, which is the study of the microbiome determinants of different drug responses between individuals. That term was coined in 2009. And that basically means just what I just said, that the recognition that microbes impact drug pharmacokinetics and pharmacodynamics, because so many of our drugs are given orally. The GI microbiome is naturally the most important. It's also the most biodiverse and metabolically active population within the host body. And this field of pharmacomicrobiomics is part of the larger field of personalized medicine, or what we would call individualized medicine, in our veterinary species, which is the idea that we can identify various features of a patient and use that to predict drug responses. And the biggest area of personalized medicine right now is pharmacogenomics, where we're looking at genetic factors influencing drug outcomes. But this is an up and coming field, understanding our everyday, our further recognition of the metabolic impact of these microbes that we're carrying around with us. And although some of these interactions that I'm going to talk about today have been identified and known about for decades, the field has really been invigorated in the past 15 years with the advent of high throughput techniques, 16S sequencing, metagenomic studies, RNA-seq, metabolomics, proteomics, which is really allowing more in-depth investigation at a systems-based level. So today I'm going to share with you some of these drug bug stories, and hopefully convince you that the microbiome has a major impact on drug levels in the body and thus on drug outcomes. So the most obvious and direct way that the microbiome can impact drugs is through that metabolic or biotransformative capacity that they have. And in eubiosis, in health, the gut microbiome can perform a lot of different reactions, metabolic reactions that can biotransform drugs, and even produce unique metabolites that the host's metabolic capacity can't produce, for better or for worse. And then, of course, as we've been talking about for the past 2 days, when we have, for one reason or another, there are negative impacts on those microbial populations. We call that dysbiosis, whether that be due disease or due to antibiotic administration. And that's going to significantly impact the metabolic capacity of the gut microbiome. In general, there's an overall reduction in metabolic capacity because there's generally a decreased overall numbers and decreased diversity. But the impact of dysbiosis is going to really be dependent on the individual drug that we're talking about, and also the individual shifts that have occurred in the microbiome population. So if you think about it in very general terms, bacterial biotransformation can kind of do one of three things to a drug. It can activate a drug. So many drugs that we administer are actually prodrugs administered in an inactive form that then have to be biotransformed into the active metabolite. And so in that situation, microbial drug biotransformation is going to enhance efficacy by increasing the concentrations of the active form of the drug. It can do the opposite. It can take an active drug and inactivate it, turn it into an inactive metabolite, and that's going to decrease drug efficacy. Or it can take either an active or inactive drug and biotransform it into a toxic metabolite, which of course is going to lead to negative drug outcomes. And these are not necessarily mutually exclusive processes. There are definitely situations, including one I'm going to show you, where the active metabolite may also be more toxic, or the inactive metabolite may be more toxic. So although I'm, putting these into these bins, recognize that it's a little bit more complex than that. So just to start off with some of our drug stories, the first one I want to tell is sulfasalazine. This is a drug that I imagine many of you are familiar with. It's used in humans to treat ulcerative colitis. We use it sometimes as a local anti-inflammatory in canine chronic enteropathy in veterinary patients. And it is a prodrug. So it does not become active until it reaches the distal gut, the colon, and bacterial reductases actually break this acyl bond and separate it into its two components. First, there's the 5-aminosalicylate or mesalazine, and this is related to aspirin. It's not absorbed, it stays in the colon, and this is what exerts the anti-inflammatory properties. But then we get this other piece of the sulfasalazine, which is sulfapyridine, which is actually a sulfonamide antibiotic. It's a very old school sulfonamide antibiotic that can be systemically absorbed and then have potentially adverse reactions like keratoconjunctivitis, Zika, or other sulfonamide-related issues. So obviously an individual's capacity for this reaction is going to impact both the potential efficacy of the drug and the potential side effects. Here's another example of a prodrug. So lovastatin, it's an anti-cholesterol drug primarily used in people, but we do occasionally use anti-cholesterol drugs in animals. And it's also a prodrug. It's activated to the active form, which is lovastatin hydroxyacid. And this is a study in rodents, basically where they were able to demonstrate that this reaction is primarily mediated by the gut microbiome. So this is looking at the reaction capacity or the reaction rate of this reaction in normal animals, animals treated with ampicillin, and then animals treated with a mix of antibiotics that were basically intended to wipe out the gut flora. And you can see here that there's a significant reduction in lovastatin activation in antibiotic-treated animals. And then, does this really matter? Yes, it does have systemic impacts. So they then took these animals in vivo and administered the drug before or after the antibiotic administration. And you can see that this is a time concentration curve, so kind of looking at overall exposure of the drug, systemic exposure of the drug. So looking at, peripheral blood samples over time, and they're looking at the active form, the hydroxy acid form. And you can see here is the normal animals, and then the animals treated with the antibiotics, and antibiotic treatment significantly reduced concentrations and the area under the curve, which is a measure of total drug exposure in antibiotic treated animals by disrupting the microbiome that is activating the lovastatin. We also said that the microbiome can inactivate drugs. A good example here is digoxin, which we don't use quite as much in veterinary medicine anymore, but it's still used for rate control in supraventricular tachycardias in small animals. And it's an important drug because it has a narrow therapeutic index. So we have to be really careful about drug levels, and anything that can impact drug levels can predispose to toxicity or lack of efficacy. So digoxin has this chemical portion called the butyrolactone ring, and there's a metabolite, an inactive metabolite called dihydrodigoxin, where basically this double bond has been reduced, and this inactivates the drug. It doesn't work anymore. Interestingly, in people, only some people have this specific inactive metabolite of digoxin, and it's related back to the presence of a reductase enzyme called the cardiac glycoside reductase, or the CGR gene, and this enzyme is actually produced by a normal gut commensal, Egerthella lenta. And all humans have this as part of their normal microbiome, but only some people carry strains of E. lenta that contain the CGR gene. So that explains that interindividual variability in this deactivation step, that only some people are colonized with strains that can do this reaction. And so that clearly is going to impact their DIG levels, whether or not you're detoxifying, inactivating DIG. And then this is where the story gets really interesting. Not only is there variability in whether or not your microbiome produces this enzyme at all, but there's actually variability in how much of the enzyme you do produce. So this is a study in mice, I believe, and here they're looking at E. lenta in culture and looking at expression of that cardiac glycoside reductase gene. And first of all, the gene, the enzyme is only expressed in the presence of digoxin. So it's an inducible enzyme, which is really interesting. But furthermore, arginine levels actually impact the expression of the gene. So when arginine is low, more gene is expressed than when arginine levels are high, meaning arginine in the environment suppresses CGR expression. So, and that's going to, again, influence DIG levels in the body, right? So then they took this into an in vivo study and looked at, again, in mice, they were looking at mice colonized with E. lenta strains that expressed CGR and E. lenta strains that didn't express CGR. And they found, first of all, the animals who were not carrying the CGR gene in their microbiome had higher DIG concentrations, which is expected, right? Because the CGR gene eliminates DIG, it metabolizes DIG away. But then they also found that the protein concentration in their diet also influenced their DIG concentrations. So here in the CGR expressing strain, there was a significant increase in DIG concentrations when dietary protein concentrations were high, but there was no significant difference, obviously, in the animals colonized with non-CGR expressing E. lenta. So this is a really interesting complex interaction where we've got, the drug, the host, the microbiome, and now the diet all influencing drug levels of this important drug with a very narrow therapeutic index. So again, going back to the concept of pharmacomicrobiomics as a part of personalized medicine, trying to understand why there's variability, interindividual variability in drug levels, the gut and its interactions with the host, the gut microbiome and its interactions with the host and the diet are really influencing those in this situation. So we talked a little, I mentioned also that drug biotransformation can also lead to toxicity. That's the case with metronidazole. There's a unique metabolite of metronidazole that's only produced by gut microbes, and it's a known carcinogen, at least in rodents. And then with chloramphenicol, which we know causes aplastic anemia, there are specific gut microbial metabolites that have been demonstrated to be actually more myelotoxic than the parent drug. So again, we modulate the immune system, modulate the gut microbiome, we may influence the differences, the amount of these toxic metabolites produced. And then an example where we've got the gut doing kind of more than one, or the gut microbiome doing more than one thing to these drugs is NSAIDs, specifically carboxyl-containing NSAIDs, which include carprofen and robenacoxib, important small animal veterinary drugs. So in this situation, we've got NSAID. The NSAID gets into the gut, it's taken orally, and then it's absorbed into portal circulation. That NSAID gets glucuronidated by the liver, and this is a detoxifying step, and it's going to enhance elimination in the bile, and then also elimination or excretion, fecal excretion of the metabolite. But the gut microbes can actually deconjugate the NSAIDs via a group of enzymes called beta-glucuronidases. They pull the glucuronide off, and that's going to prolong the exposure of NSAIDs because it's going to allow that parent drug, its reactivation, so that parent drug can then be reabsorbed, and we wind up getting enterohepatic recirculation, similar to what we see with bile acids. So that's going to increase systemic exposure, which is going to potentially increase efficacy, but it's that parent drug that gets converted by other enzymes to potentially toxic metabolites that are thought to lead to or mediate a lot of the adverse drug reactions that we see, the GI upset, et cetera. So this is a situation where gut biotransformation of the drug increases both efficacy and toxicity. And then to further investigate this process in models where antibiotic induced dysbiosis occurs, that's going to reduce the glucuronidase capacity. There's just fewer microbes around to express those glucuronidase enzymes. So we get less reactivation of the NSAID, which is going to decrease toxicity, which is a good thing, right? Because we're going to facilitate elimination of the NSAID rather than allowing it to stick around in the gut longer. But then that's also going to decrease systemic exposure because we've reduced that enterohepatic recirculation and decreased the amount of NSAID that's available to make it into systemic circulation and have its effects. So in addition to directly biotransforming drugs, the gut microbiome can also indirectly influence how the patient handles those drugs, specifically looking at both absorption and then host metabolism or biotransformation of the drug. So here's an example of a study looking at amiodarone, where they showed that pre-administration of an E. coli probiotic actually increased drug exposure in systemic drug exposure, and this was thought to be due to the probiotic enhancing drug absorption from the gut. And they didn't really, at least in this study, specifically identify a mechanism by which that happened, but they hypothesized a few different mechanisms could be present, potentially an impact of the shifts in the microbiome on gut pH, or potentially gut motility, which can both impact how much of a drug gets absorbed. They also hypothesized that the probiotic could alter transporter expression or even enterocyte drug metabolism. And I think that's something that not a lot of people always directly think about, but the drug metabolizing enzymes are not just expressed in the liver; they're also expressed in the enterocytes, which is an important modulator of drug absorption, of how much drug eventually gets to systemic circulation. And so these two features have actually very recently been evaluated in a very new study out of Drug Metabolism and Disposition, which is one of the major pharmacology journals. And what they did here was, again, working in rodents and rats, they administered a probiotic. And first, just to prove to everybody that the probiotic actually did something, they did 16S sequencing and demonstrated that there were changes in the population structure, changes in alpha diversity and beta diversity. But then they took jejunal samples, jejunal enterocytes, and actually did RNA-seq on them, looking at drug metabolizing enzymes and drug transporters. And they did indeed find that the probiotics significantly changed jejunal expression of all of these things. So phase 1 enzymes, these are primarily our cytochrome P450 enzymes. They tend to be like oxidating or hydroxylating enzymes. And not completely across the board, but in general, this specific probiotic actually tended to decrease expression of the CYP enzymes. It also altered expression of a lot of the phase 2 enzymes. These are drug conjugating enzymes. So things like glutathione S transferases, N-acetyltransferases, sulfonylureal transferases, glucuronidating enzymes, basically enzymes that pop a small molecular motif like a sulfate or something onto the drug. That's a big part of detoxification. And there was kind of mixed results here. Some of the enzymes were upregulated, some of the phase 2 enzymes were downregulated. And then they also looked at transporter expression. Most of these, the SLC, that's a solute carrier. So those are transporters. They may be influx or efflux transporters. A lot of gut transporters that work on drugs are efflux transporters to basically trying to protect the systemic circulation from xenobiotics. And a lot of these, at least again with the particular probiotic that they used, actually caused decreased expression of those transporters. So really interesting stuff. I think I'm running a little low on time, but I'm going to skip over this. But just to say that in this same study, they looked at a bunch of different probe drugs and actually demonstrated a significant difference on systemic exposure of these drugs just by administration of a probiotic. And then to kind of bring it all back together, to take it all the way through, the process of absorption, distribution, and elimination, the gut microbes can actually also impact hepatic metabolism through a couple of different mechanisms. One is via competition. So this is a story about acetaminophen. We all know acetaminophen, which is actually metabolized by a few different enzyme classes. It can get sulfated, it can get glucuronidated, and then it can get oxidized. Sulfated and glucuronidated metabolites are inactive, whereas oxidative metabolites are toxic. That's where NAPQI comes from, and acetaminophen hepatotoxicity. And acetaminophen is preferentially sulfated and converted into these inactive metabolites. But when there's decreased sulfation or glucuronidation capacity, acetaminophen will get pushed towards oxidation. The CYPs will get recruited. So there's this compound called picresol, which is a endogenous metabolite produced by primarily Clostridium in the gut, and this gets systemically absorbed. And if you take a look at it, it actually looks very structurally similar to acetaminophen, and that's important because they have similar substrate preferences when it comes to post-hepatic metabolism, that picresol is preferentially metabolized by sulfation and then glucuronidation. And what this can potentially do is, because there's increased competition for these sulfating and glucuronidating enzymes, it can actually push acetaminophen metabolism towards the recruited enzymes, the less preferred enzymes, which actually may increase the production of toxic metabolites. And this has been looked at in people where they basically just measured the amount of picresol that people have in their bodies, and then looked at the sulfation to glucuronidation ratio of acetaminophen. And you can see there's an inverse relationship here, that the more a person makes or metabolizes picresol, the more the acetaminophen metabolized is shifted down to the glucuronidation. And this is, again, I know I'm running out of time here, but just to bring this up, that in general, there's this concept that we really need our gut microbiomes to basically train our own hepatic metabolism in handling xenobiotics. And this is primarily done through the production of unique bacterial metabolites like secondary bile acids, also the indoles that Patrick was talking about, are all produced by the gut, but then they get absorbed and the liver has to metabolize that. And so that actually induces and basically trains the liver to produce those xenobiotic metabolizing enzymes like the CYPs, which we classically think of as drug metabolizing enzymes, but they're not. They're not just there for drugs. They're for a lot of different xenobiotics, many of which are coming from the gut. So just thinking forward, how might we use this in the future? And, a lot more work is done before we start implementing any of these things. But a lot of the therapies that we've already been talking about over the past 2 days have the potential to influence drug metabolism and drug disposition. Anything that can alter the gut microbial composition can also alter their biotransformative capacity and thus influence drug pharmacokinetics. We've shown with the digoxin example, we know that dietary therapy will alter drug or microbial composition, but through, like we just showed in that digoxin example, that the dietary therapy can actually directly alter the biotransformative capacity of the gut microbiome. And then I think Dr. Simpson and Dr. Barco have both talked about this a little bit, but there's this idea that we can actually use compounds to directly modulate microbial biotransformation in the gut. So what I showed you with the NSAIDs and the antibiotics decreasing systemic exposure, that same thing has been demonstrated to the same effect. Instead of wiping out the microbiome, they just use the beta-glucuronidase inhibitors, these small molecules that specifically inhibit a microbial-derived enzyme, and showed the same impact. So hopefully, through this talk, I've convinced you that the gut microbiome is potentially really important in changing how drugs are handled by the body, both by direct and indirect effects, that it's potentially an important contribution in an individual to variation. And then just a reminder, taking it all back to the beginning, that although we know the microbiome can impact drug metabolism, drugs also impact the microbiome, so this is a really complex system, and we need to account for both of these processes when we're thinking about it. And we're living in a new age where we have the tools to really investigate these processes. So with that, I can take any questions.